Earlier today, I started this lecture, and as I said, I wanted to get the fluorescent sperm identification done before we got into the room so y'all can have some idea about what you were looking at. But the other part of the advances in the microscopy is the fact that we can now do other stuff with fluorescence, especially what we cannot do in forensics now is diploid cell mixtures. If we have a male-female mixture on a bite mark or a neck swab or fingernail scrapings, there's really no way to separate it. You just basically do your extraction and do your quant amp and go with what you get. And some of the problems with that, although my slide seems to be moving by itself, is initially a sample may not be identified as a mixture prior to analysis. And well, I've seen this before is they take a swab from a chest or a victim's neck. That may actually contain semen as opposed to two epithelial cells mixtures. You don't know that. You just assume, okay, next swab is going to be two sets of buccal cells or some type of skin cells and buccal cells. It may actually be a semen-containing sample as well as a mixture of the victim. You don't know that unless maybe you've read the history or something like that. So this is a point where microscopic identification might help you to say, hey, this is something we need to do. Maybe we can do it with sperm ID, or maybe it is a diploid cell mixture. The mixture may not be apparent because there is a preponderance of DNA from one of the victim in most instances. If you do like some swabs, a bite mark or a breast swab may have or fingernail scrapings may have a preponderance of victim's type. So much so that you never see the little blip of the Y chromosome, or maybe all you see is a blip of a Y chromosome. Hey, this is a very small, maybe a greater than 20-to-1 mixture of one person over the other. And remember I talked about microscopy as a very sensitive way of finding something small in a large population of other cells. So again, if you look at this, if you ran the fingernail scrapings, you may never see the Y part in the amplification. You may never know it's a mixture. If it's a 20 or 30 or 50-to-1 mixture, you may never see the small minor component. Also, with diploids, basic mixtures, you have the interpretation issues. I think Christine and Kelly both talked about a little bit. How do you interpret when you've got equal amounts of one or smaller or much smaller amount of another one? And right now, there's no current way of separating diploid cell mixtures by the forensic techniques we have. We look at the available kit. It's called chromosome paint, or it's by Weiss, this is the CEP. We look at this stain here. This is what's called FISH, fluorescent in situ hybridization. You have a nuclear probe or nucleic acid probe, which can be hybridized to various chromosomes. You can have all sorts of probes that will identify different types of genetic areas. Most of these genetic areas are associated with some type of disease or genetic mutation or something like that. However, there is one that's sold that's used for X and Y, identifying the X and Y chromosomes. This is sold to identify bone marrow transplants, primarily the repopulation of the minor or the recipient in a different sex bone marrow transplant. That's how you would use this. This is, again, FISH. It's got the X and Y probes. It's used for interphase nuclei. You don't have to, as you used to years ago, go to the metaphase. Use something like colchicine to stop them at the metaphase point, but this is used for interphase nuclei. And this is actually a new capability. This is something that cannot be done currently in the laboratory, but this microscope, the LMD microscope, with the fluorescence capabilities, adds this capability. This can be done. We have done it on forensic samples. You can look up there. If you see at the top, here's y'all got to see the DAPI stain today where your nuclei are covered. Here's the same two slides at the bottom, or same two cells at the bottom. They happen to be both male cells. If you look in there, you see the bright green. Let's see. The bright green, that's your Y chromosome. And up here is the red dot. That is the X chromosome. And on here, here's your Y, and here's your X. Those are lymphocytes. They stain quite easily. This was done a little over a week ago. It's not a hard procedure. My daughter did it. That was the first time she'd ever done it. It came right up first time. Really nice, good stain. Now, lymphocytes are pretty easy. I have to say that. And these weren't necessarily forensic type samples, but it shows you what is the optimum that you can look at. And this was the old microscope. I think the new one probably does a better job than this one. It can be a little bit brighter or something like that. But this came right up. Very easy to identify your male cells. Now, your female cells are going to have the 2 red dots, the 2 X chromosomes. Here's actual buccal samples taken from dried swabs, extracted from a cotton swab, and plated and run on there. Here you can see the Y chromosome and the X chromosome, and down there the female cell that has the 2 X chromosomes. This is not quite as nice as clean and cut as the other one, and that's one of the issues with this type of staining. But this is a forensic type sample. This is a sample that came off a dried cotton swab. It was a mixture of male and female cells. We put them all together, and hey, it works. It can be done. Can it be improved? Yes. But remember, we're right now in this type of testing, we're in the infancy. We're one of the few people, actually, I think, in the world that have done something like this on forensic type samples. I know other places have done it, and I think Britain has done where they've really put in a lot of money into it. But this is one of the first slides. This slide is actually about a year and a half old. This was done a long time ago. And it does work. So this is a technology we see. It will work, it can work, and it works on forensic samples. It needs to be optimized, it needs to be improved. It's kind of like what Kelly started. When she first started, she was getting ups and downs and not really good stuff, but as she progressed through it, everything got reproducible, came up nice and fine. This is still in infancy. It can be done. It's got some reproducibility, but there are still some problems with it. One of them is I think we need a little bit stronger probes. Is there a concern with this? Okay, we're putting Y chromosome DNA in there as a Y probe. Here's a Y STR profile from those stained slides, from those, not those particular cells, but from a slide that's been stained by fish with the X and Y probes. Gets a nice Y profile. So this method does not affect the Y chromosome testing or Y STR testing. That's always an important concern. Is that an issue? Yes, it would be an issue, but here we have a nice clean profile. Actually, probably not a full profile, but it's pretty well up there. Extreme cell mixtures. We've looked at 1 to 99, 1% male cells all the way up to 1% female cells. Number of loci in the male profile, the first one you see is a 99 to 1. That means we made a mixture of cells, 99 female cells to 1 cell, 1 male cell. We looked at the smear and we were able to identify several cells, and we got 7 loci in the male profile, 12 in the female. As it went on down, we got different proportions, but we were able to find male cells or the opposite sex cells even as little as 1 to 100 mixtures. Real case results. The real meat of this matter is, can this work on a real case? And as Kelly was talking about, we have a number of non-probative rape kits. We probably have, 30 to 50 kits they've given us that they've closed out. These are sitting in the evidence locker. I know there was a thing about, what about degraded DNA? Well, some of these kits are 3, 4, 5 years old. They've been sitting at room temperature. So we've got a really good base of non-probative sexual assault cases to work with. And we don't know the results of them. We haven't actually taken them out and tried running them yet. They were closed out before we even got to the case. So we took a sprinkling of these samples, or sprinkling of these cases, and this was run as part of one of the analyst's master's thesis on this. Identification and dissection of sperm cells well established. In other words, you've looked at Christina. I've heard Kelly and Christina talk about, yeah, we can do sperm identification. They can cut them out. They can amplify them. That's real world right now being able to be done. Can we do this fish on those? Can we do this X and Y chromosome or chromosome paint to identify these male and female cells on real cases? Now, these following results I'm going to give you are based upon diploid cell analysis. We weren't looking for sperm in these cases. We weren't trying to identify sperm. No sperm were identified. Fish doesn't work on sperm. The DNA in there is too condensed for the hybridization to occur. So every cell that was identified in there was a diploid cell. It was not a spermatozoa. In this instance, 40% of the cases had at least one sample with enough diploid cells to obtain a male profile. We obtained these diploid cells from vaginal swabs, bite marks, and fingernail scrapings. All samples taken as part of a rape examination, a typical rape examination. They were all collected by sane nurses during their examination. At the time, they were collected as if they were a regular rape case. There was no prior indication that these cases would ever be used for research. We eventually got the ability or orders that these cases could be discarded, or these kits could be discarded or otherwise not analyzed. We obtained permission and put them into our research-only stack. So there's nothing in these cases, there's not a samples that were taken for the intent of doing research on. They were taken as part of real case analysis or real case workups. In number one, we had the fingernail scrapings. Here we see the victim's type on the left-hand side. Fingernail scraping where we got 30 female cells. We got a pretty decent profile there, pretty much a full profile. In the fingernail scrapings, we were able to identify 15 male cells. And I believe this was actually done before we got a final optimization of that you've worked with. This was done with Identifiler before Kelly had developed a final optimized procedure. So some of the work here is not quite as this is probably would get better results now. Okay, here we got 15 male cells. We do get a partial profile. Again, the suspect is on the far right-hand side of the known. We do get a lot of haploid types or a lot of single types. But what's important is, in every instance here, that you did have the type that was obtained was characteristic or found within the suspect's type. The vaginal swab, we actually identified 25 male cells, not spermatozoa, but male diploid cells from the vaginal swab in this sample. We were able to get some get profiles out of there, but we did get a few other types that came in there. These other types, I believe, were characteristic of the victim. But nevertheless, from a vaginal swab, we were able to obtain diploid cells that came from the male. Just think where this would be important. Vasectomies. Right now, what can you do with a vasectomized individual? Not a whole lot, is it? You basically have to deal with a mixture. And if you have a large amount of female epithelium, you may not ever see those male cells in there. Or you may be able to fish them out, fish, no pun intended, with the Y-filer, if you can recognize that you need to. But here you have the capability of going into a vaginal swab and examining it for Y cells. Now, there's no doubt that this is a little bit more extensive technique than the sperm highlighter or sperm paint, but it's something that can be done. It's something that I think will be available within a short period of time, not quite as quickly as the sperm ID and the sperm highlighter and things like that, but this is a technique that can be done. It's applicable to a lot of sexual assault cases where we're now having to deal with mixtures. Here's the vaginal swab. Case number 2. Okay, the victims, we have the known type there. We have the vaginal swab. 20 male cells collected from the vaginal swab. Again, these are not spermatozoa. With this, we've gotten almost a full profile from the suspect. And here's the vaginal swab. Here's what we got when we did actual digestion of it. And you can see it was a wonderful mixture. At least it showed most of the profiles of both people together. But one thing about this vaginal swab where we did the fish or the XY chromosome paint, we were able to get a pretty decent profile out of that. Again, this was done before we've optimized the identifier amplification. Bite mark swabs. Here we have the bite mark swab, and this was the profile obtained with 7 male cells. The bite mark swab was actually run, and there's the profile that was gotten, that was obtained, compared to the profile we got from the 7 male cells. We also got the vaginal swab. We got female cells, and here's the suspect's type, and you can compare that to what we got for the bite mark swab. Yes, there is dropout, but remember, we're only 7 cells there. All of these samples, all the profiles that were obtained in this sample were run against, we put it in a local coded search as is. Obviously, those profiles are not available or not capable of running against the national level. There's just too much dropout for them, not enough loci, too much profiles, or the profiles weren't of adequate quality. However, on a local search, every one of those samples, every one of those actual samples had one hit, and it was to the person associated with the case. They were checked against a couple of thousand or so samples in our local database, and in every instance, those profiles that we obtained, even as limited as they were, matched to the individual, and only one individual in the database, and it was the one that was part of that case. And here's the acknowledgments. Jennifer and Kelly had done a lot of work on this, the crime lab for letting us have fun, and LSU Health Science Center, which actually was where Jennifer did some of her master's thesis work on this type of analysis. So if there's any questions right now on this, I will.
How long does the fish protocol take?
Fish protocol, you have to have your chemicals made up to start with. Some of them have to be fresh. It takes about 30 minutes the first day. It goes overnight hybridization, and about an hour the second day. And these are, as Andy talked about when we looked at the microscope, these are much more susceptible to bleaching than the sperm highlighter. So you do have to examine them right away. It's not something you're going to be able to examine and then put them away and then come back, days later. Although they say the slides can be frozen and still re-examined for up to 12 months.
This requires a very good fluorescence microscope.
Yes. You saw the buccal, the first ones I did on lymphocytes, those were as is. Those I just took the picture right off the microscope and put it up there, and you can see that worked really, really nice. Y'all, I don't know if y'all in the back could see the X and Y chromosome. Mm-hmm. That's pretty good. The buccal cells that we used, those I had to kind of enhance the color, like go into Photoshop and pull up a little bit of the color, brighten it up a little bit. They were seeable, but they don't make good display photographs.
The reason for
That is that the camera that's on the microscopes now, and most cameras that are on these kinds of systems, are just not optimized for fluorescence microscopy. Okay, we're using this camera that's normally a bright field camera for fluorescence, and it's not optimal. The camera that Pat has can do some decent fluorescence, but in general, in the research arena that are doing fish, they're using digital cameras that are cooled cameras for low noise and all this kind of stuff. So you've got to know that. Okay, is this system capable of doing that? Yes. What we have here is not that system. So I just want to say, in general, any fish technology that you want to go after, you need a good microscope, then you need a very decent camera. The image quality can be much greatly increased, but realizing what you're doing, live image and all this kind of stuff. Again, the exposure times typically are going to be much longer than we even saw here. But again, using a cool digital CCD camera. The cameras themselves are going to cost in the neighborhood around anywhere from $10,000, $20,000, $30,000. And they typically are black and white cameras, and you assign the colors after the fact. That's in research. I mean, for some of you, black and white cameras are more sensitive.
But the microscope we had was capable of doing that work on actual casework type samples. Yes, the 99-to-1 mixtures, we made those up, but those other things, those casework, those were actual casework samples, and they worked quite well. I mean, the results were, it was really shocking to me how well the results came up.
And honestly, I would be curious to see what those would do with the optimized identifier.
Right.
They still might not be like perfect or full profiles, but I don't know. I'd be curious to see.
Any other questions here? Okay, I think Christine want to talk. Oh,
Yeah, I just want to say one thing before Pat continues on. We're supposed to look at our data. Unfortunately, we've been kind of troubleshooting today, and the data is very poor. We're trying to figure out actually what's going on. It looks like most of you either had no results, or if there was some results, it was just one, a couple peaks. And then there was just one that also was really low. That was just a mixture. So it's data that I wouldn't expect given the number of samples we collected. Now, obviously, because we were introducing these protocols in a completely new facility, and we're trying to figure out what went wrong. So what I'm going to do is I'm kind of going to look at the data some more, and I have all your email addresses too. And I'm hoping that I can get this solved before the next course, which starts Thursday, and then come up with some explanation why it didn't work. And then hopefully if the next class comes in and we get good data, I can send that to you so you at least get a feeling of what your data might look like. So I'm sorry for the disappointment on that. Trying to see whether it had anything to do with the collection or the lysis or something like, if something was different than what we normally would do in our protocols.